Hey everybody, Kyle here, giving you a quick introduction to Chapter 3. Chapter 3 in our textbook for microbiology is called Cells and Methods to Observe Them, so cells and microscopes is going to be your main focus here. We'll look through the chapters here, and then I'll let you go to watch some videos to recover most of, hopefully, your information that you learned back in biology. Get those things top of mind before you dive into the textbook itself. The different sections of this textbook are going to really try to point out how things are different, new, and important for prokaryotic life forms or for our single cellular microscopic eukaryotic life forms. So big things to look out for is how is this different than what you saw in general biology. How they added another layer of information, more than just saying plants have cell walls and animals don't, yay. Plants make it out of cellulose. They're now going to say, okay, bacteria also have cell walls, and it's mostly made out of dextran, and many other complex things we're also going to learn about. And fungi have cell walls, but it's made out of chitin, and so things like that. They're going to just add an extra layer of information more than just what is a cell wall. Our different things here. First one will be prokaryotic cell structures and their functions. We'll talk about it as an envelope to encompass all aspects of what encloses our cell. And how is that more and different in prokaryotic life forms. 2, again, definitely look at these learning outcomes and use these as your study guide as well. Make sure by the time you reach the end of this chapter, or definitely by the time you reach the end of the lecture, you feel pretty comfortable with these. So we're going to look at different components of the cell wall of prokaryotes. How are they different? How can we use them for both staining to determine different microorganisms? And how do we use them in drugs to target and kill these without killing the host we're putting the drug into? Structures outside the cell wall of prokaryotic cells. This is not just a prokaryotic thing. Plants often do this. Even we have layers of secretions we release into our lungs, through our digestive tract. But we're going to call special attention to these within prokaryotic cells because they are so small and they have to survive in often very hostile environments that are either trying to attack them or are not very conducive to life. So they have a lot of special things in these capsules and slime layers. 4 internal components of prokaryotic cells. This is going to be, okay, great, these prokaryotic cells can do everything a eukaryotic cell can do, but they don't have internal membrane-bound compartments. So what do we call these areas? How do they still get these things done? How do they organize themselves? We'll jump into eukaryotic cells. Remember, fungi, algae, and protozoa are eukaryotic microorganisms we need to know. So don't think of it as just animals and plants. We're going to look at all the different structures, all the membrane-bound compartments, and you're going to need to relearn, if you don't still have them top of memory, what the major functions of these compartments are. We're going to focus on protein structures. We looked at in class today how proteins are the major workforce for a cell and the major structural components for a cell. So this chapter is going to look a little more detailed into that, including for movement, the flagella and cilia. Here is the rest of the information on the membrane-bound organelles, where they'll really dive into the nitty-gritty of each organelle. Then we get into microscopes, light microscopes, electron microscopes, scanning probe microscopes. What's the terminology we use when we talk about the ability to blow something up and see it versus be able to see the difference between things. We will use light microscopes in this lab, no electron or scanning probe, but we will use them at a higher magnification resolution than you've used before with the oil immersion lens. The next and last is how to prepare specimens for light microscopes. I'm sure you've done wet mounts before in your biology lab, but we're going to want to look at specifically how to use these different staining techniques to help identify. So you'll have learned how those stains apply to cell walls earlier in the book. Now you're going to look at how do we actually use this in the lab, and we will use that in our own lab here because because that's where I still am. Good luck, everyone. That is chapter 3. Come to class with any questions you got, and we'll try to rehash and focus on the most important aspects of what you read and prepare for. Remember to watch those videos and do the asynchronous assignment on D2L that links you over to McGraw-Hill Connect. Take care, everyone. Bye.